Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with regeneration, the science of animals that can regrow anything. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. This realm unfolds with remarkable diversity, showcasing the unique adaptations that various species have developed over millennia. Regeneration serves as a vital survival mechanism, allowing certain animals to recover from injury or loss, often with astonishing efficiency. Among the most notable are species like the axolotl, a salamander capable of regrowing limbs, spinal cord, and even parts of its heart and brain. This remarkable capability arises from specialized cells that can revert to a more primitive state, enabling them to differentiate into various tissues. Equally intriguing is the planarian, a flatworm that can regenerate its entire body from just a fragment. This organism harnesses a robust population of pluripotent stem cells, which play a crucial role in its regenerative processes. Observations have shown that these cells mobilize rapidly to form new tissues, illustrating the underlying biological mechanisms driving regeneration. The sea star presents another fascinating case. When subjected to predation, it can lose an arm, which, given the right conditions, will regenerate fully. This process is not merely a physical restoration. It also involves complex signaling pathways and cellular responses that ensure the new limb is functional and integrated into the organism's body plan. These examples highlight the intricate balance of biology and environment, revealing the underlying processes that govern regeneration in the animal kingdom. Early observations of regenerative species trace back to ancient civilizations where accounts of extraordinary recovery sparked curiosity and wonder. The Greeks, for instance, documented instances of lizards growing back their tails, attributing this phenomenon to divine intervention. Observations such as these laid the groundwork for subsequent inquiries into the mechanisms behind regeneration. In the 17th century, naturalists began to formalize their studies. Remarkable adaptations were noted in species such as the axolotl and planarian, with their regenerative capabilities prompting rigorous scrutiny. These inquiries highlighted not only the astonishing ability to regrow lost body parts, but also the potential for understanding broader biological principles. The 19th century ushered in a systematic approach to regenerative research. Researchers like Johann Wolfgang von Goethe and later Thomas Huxley explored the cellular and anatomical structures involved in regeneration. Their meticulous observations revealed that regeneration was not a mere anomaly, but rather a fundamental biological process shared among various species. This scientific pursuit continued into the 20th century, as advancements in microscopy and experimental techniques allowed for deeper exploration of the cellular dynamics at play. These observations enriched the understanding of regeneration, paving the way for contemporary research that seeks to unravel the genetic and molecular foundations of this extraordinary ability. The journey from myth to empirical investigation illustrates humanity's enduring fascination with the resilience of life. This enduring fascination has led to significant advancements in understanding the evolutionary mechanisms behind regeneration. Various species exhibit remarkable regenerative abilities, showcasing diverse biological adaptations. For instance, Certain amphibians possess the capacity to regenerate limbs, tails, and even parts of their hearts, fundamentally altering the course of their life cycles. The cellular processes involved in regeneration often hinge on the activation of specific genes and the mobilization of stem cells. This phenomenon is particularly evident in species such as salamanders, where the regeneration process begins with the formation of a blastema, a mass of cells that can differentiate into various tissues. This region serves as a critical hub for regeneration, highlighting the intricate relationship between cellular signaling and tissue formation. In contrast, other organisms, such as planarians, exhibit an extraordinary ability to regenerate nearly all body parts due to a high concentration of pluripotent stem cells. This characteristic allows them to restore their entire anatomy after significant injury. The evolutionary pathways that have shaped these abilities reveal a complex interplay between environmental pressures and genetic predispositions. Research into these regenerative processes not only deepens the understanding of biological resilience, but also holds potential implications for regenerative medicine in humans. Insights drawn from these remarkable creatures may one day inform new therapies for conditions involving tissue loss or damage. Regeneration encompasses a variety of biological processes, allowing organisms to restore lost or damaged structures. Two primary types of regeneration are observed: complete regeneration and partial regeneration. Complete regeneration involves the restoration of an entire organism, as seen in species such as the axolotl, which can regrow entire limbs, spinal cord sections, and even parts of the heart. Partial regeneration, on the other hand, may result in the regrowth of specific tissues or organs, as seen in certain species of starfish, which can regenerate lost arms. Mechanisms of regeneration vary significantly among species. In some organisms, such as salamanders, specialized cells known as blastomal cells play a crucial role. These cells are capable of dividing and differentiating into various cell types needed for regrowth. In contrast, planarians utilize a population of pluripotent stem cells that can transform into any cell type required for regeneration, highlighting the diversity of regenerative strategies. Molecular signaling pathways are also integral to the regeneration process. Growth factors and genes regulate cellular activities, ensuring that regeneration occurs in a precise, orderly manner. This intricate interplay of cellular and molecular events enables organisms to execute repair and renewal effectively. Understanding these mechanisms not only enhances knowledge of biology, but also opens avenues for medical advancements in regenerative medicine. The role of stem cells in regenerative processes is pivotal, serving as the foundational elements that drive healing and tissue renewal. These undifferentiated cells possess the unique ability to transform into various specialized cell types, providing the necessary building blocks for regeneration. In specific organisms, such as salamanders, stem cells migrate to sites of injury, efficiently orchestrating the regrowth of limbs and other complex structures. This remarkable ability hinges on several factors, including the signaling molecules that guide stem cell activity. These molecules create an intricate communication network that directs stem cells to activate and proliferate in response to damage. The environment surrounding these cells, often referred to as the stem cell niche, plays a crucial role in determining their fate and function. In mammals, stem cells are primarily located in specific tissues, such as bone marrow and skin, where they contribute to ongoing maintenance and repair. While their regenerative capabilities are limited compared to some other species, advancements in stem cell research are beginning to unlock new potential. Scientists are exploring methods to harness these cells for therapeutic purposes, aiming to treat conditions ranging from heart disease to neurodegenerative disorders. The exploration of stem cells continues to deepen our understanding of biological regeneration, expanding the boundaries of what is possible in medicine and biology. Comparative anatomy reveals the diverse regenerative capabilities present across various species. In this region of study, organisms such as salamanders and axolotls stand out for their remarkable ability to regenerate limbs, tails, and even parts of their hearts. These amphibians utilize a combination of stem cells and specialized tissues, allowing them to restore complex structures with precision. Other species exhibit unique strategies for regeneration. Sea stars possess the ability to regenerate entire arms, and in some cases, a severed arm can develop into a new individual. This remarkable process relies on the presence of undifferentiated cells that can transform into the necessary tissue types. Similarly, certain species of flatworms known as planarians can regenerate their entire bodies from small fragments, showcasing an extraordinary level of cellular plasticity. Invertebrates like the hydra display yet another form of regeneration, continuously renewing their cells throughout their lifespan. This ability is linked to a unique population of stem cells that persist, allowing for constant growth and repair. The regenerative processes observed in these organisms raise profound questions about the evolutionary pressures that shape such capabilities. Understanding these various mechanisms across species not only enhances biological knowledge, but also holds potential implications for human medicine, potentially inspiring new approaches to healing and regeneration in higher vertebrates, including humans. The planarian serves as a quintessential model organism for studies of regeneration, exhibiting remarkable cellular plasticity. This flatworm possesses the unique ability to regenerate entire body parts, including its head and tail, from mere fragments. The underlying mechanisms involve a population of pluripotent stem cells, known as neoblasts, which are distributed throughout the organism. When injury occurs, these cells migrate to the wound site, proliferating and differentiating into the necessary cell types to restore lost structures. Investigations into the genetic and molecular pathways governing this regeneration reveal a complex interplay of signaling molecules and transcription factors. Notable among these are the want and hedgehog signaling pathways, which play critical roles in the regulation of cell fate during regeneration. This region of study not only uncovers fundamental biological principles, but also highlights the evolutionary significance of regenerative capabilities. Planarians exhibit a high degree of regenerative efficiency, making them ideal subjects for understanding the cellular and genetic frameworks involved. The insights gained from these organisms may illuminate pathways that could be harnessed or mimicked in higher vertebrates by exploring the regenerative capabilities of the planarian. Researchers aim to unlock potential applications in regenerative medicine, offering hope for future advancements in healing damaged tissues and organs in humans. The primary cells responsible for regeneration in planarians are known as neoblasts. These pluripotent stem cells possess the remarkable ability to differentiate into various types of tissues. Their presence throughout the planarian's body allows for a rapid and efficient response to injury. When a planarian sustains damage, neoblasts migrate to the site of injury, proliferating and initiating the complex process of regeneration. This region becomes a hub of cellular activity. Neoblasts rapidly divide, producing a diverse array of specialized cells that contribute to the formation of new tissues. The precise coordination of signaling pathways governs this process, ensuring that the appropriate cell types emerge at the right moment. Factors such as Wnt, hedgehog, and Notch play pivotal roles in regulating neoblast behavior, guiding their differentiation and movement. The regeneration process also involves the intricate interplay between neoblasts and surrounding tissues. As new cells form, they integrate into the existing structure, restoring functionality. This dynamic interaction underscores the importance of the cellular environment in successful regeneration. Research continues to delve into the molecular and genetic mechanisms underpinning neoblast activity. Understanding these processes not only illuminates the capabilities of planarians, but may also reveal insights applicable to broader fields, including human regenerative therapies and tissue engineering. Axolotls stand out among regenerative marvels within the salamander family. Their ability to regenerate limbs, spinal cord, heart, and even parts of the brain showcases an extraordinary biological mechanism. This region of the animal kingdom offers unparalleled insight into regenerative biology, as these creatures retain juvenile features throughout their lives, a condition known as neoteny. This trait allows them to maintain a potent regenerative capability, unlike their terrestrial relatives. Upon losing a limb, axolotls initiate an intricate process. Cells at the injury site transform into a mass of undifferentiated cells, forming a structure known as a blastema. This blastema serves as a reservoir of potential, allowing for the precise replication of lost structures. Signals within the tissue guide the differentiation of these cells into the required cell types, resulting in the restoration of the limb with remarkable accuracy. Research into axolotl regeneration has revealed the importance of specific genes and molecular pathways. These mechanisms orchestrate complex cellular behaviors, allowing for the seamless integration of new tissues with existing structures. Understanding these biological principles may pave the way for advancements in regenerative medicine, offering hope for therapies that enhance healing in humans. The exploration of this remarkable creature continues to uncover the secrets of regeneration, highlighting the intricate interplay of biology at work. Limb regeneration in axolotls showcases a unique biological process that has fascinated scientists for decades. This species, native to the lakes of Mexico, possesses the extraordinary ability to regrow lost limbs with remarkable precision. Following an amputation, the initial response involves the formation of a blastema, a mass of cells that will eventually develop into the new limb. These cells, derived from various tissues, undergo a process of de-differentiation, reverting to a more primitive state, which allows them to proliferate and subsequently differentiate into the necessary structures. The regeneration process is characterized by a series of orchestrated steps. Starting with wound healing, specialized cells migrate to the injury site, forming a protective layer over the wound. This layer facilitates the transition from a healing response to regeneration. The subsequent assembly of different cell types is crucial as they work together to recreate muscles, nerves, and cartilage. Molecular signals play an essential role in this process, guiding the cells toward their appropriate fates. Research into the axolotl's regenerative capabilities has implications that extend beyond mere curiosity. Understanding the mechanisms at play may provide insights into potential applications for regenerative medicine in humans. The study of this remarkable organism continues to bridge the gap between evolutionary biology and advanced medical research, offering a glimpse into the potential of regeneration across species. The wound epithelium plays a pivotal role in limb regeneration, acting as a protective barrier while orchestrating a myriad of cellular processes. This layer forms rapidly after injury, creating an environment conducive to healing. Its formation marks the initiation of regeneration, signaling the underlying tissues to mobilize resources for recovery. Cells within this epithelium begin to secrete growth factors and signaling molecules, guiding the behavior of nearby progenitor cells. These progenitor cells, often dormant, receive cues from the wound epithelium, prompting them to enter a proliferative phase. This activation is crucial as it leads to the formation of a blastema, a mass of cells capable of differentiating into various tissue types necessary for limb reconstitution. The wound epithelium not only protects the exposed area but also synthesizes extracellular matrix components, providing structural support for the regenerating limb. An intricate communication network emerges between the epithelium and mesenchymal tissues, facilitating the spatial organization and patterning of the new limb. Research indicates that this communication is highly conserved across species, highlighting evolutionary strategies for regeneration. Understanding these mechanisms deepens insight into regenerative capabilities, with implications for medical therapies aimed at enhancing healing processes in humans. The regenerative potential inherent within the wound epithelium emphasizes the intricate balance between injury management and tissue restoration. The blastema serves as a crucial component in the process of regenerative growth. This specialized mass of cells forms at the site of injury, acting as a reservoir of undifferentiated cells ready to transform into various tissue types. Within this region, cellular reprogramming occurs, enabling the recruitment of stem cells and the activation of growth factors essential for regeneration. In certain species, such as salamanders, the blastema facilitates remarkable regeneration of limbs. Following amputation, a complex sequence of cellular events initiates, leading to the proliferation of these undifferentiated cells. They aggregate to create a blastema, which then differentiates into muscle, bone, cartilage, and skin. This process exemplifies the remarkable adaptability of biological systems in response to damage. The presence of specific signaling pathways within the blastema further underscores its significance. Molecular cues guide the cells, determining their fate and ensuring the restoration of the original structure. This region must maintain a precise environment where gradients of growth factors orchestrate cellular behavior and organization. Studying the mechanisms governing the blastema can unlock potential applications in regenerative medicine. Understanding how specific species harness these biological processes may pave the way for advancements in human tissue repair, offering insights into enhancing healing capabilities and regenerating damaged organs. The intricate workings of the blastema remain a focal point in the exploration of regenerative biology. Regeneration in echinoderms, particularly sea stars, showcases a remarkable biological phenomenon. When subjected to predation or environmental stress, these creatures can detach and regrow lost arms. This process initiates with the formation of a regeneration blastema, a mass of cells that will eventually differentiate into the various tissues required for a fully functional arm. It begins with a complex interplay of cellular signaling and gene expression, ensuring that the newly formed limb mirrors its predecessor in both structure and function. The regeneration process can vary significantly among species. In some instances, the entire arm can regenerate, while in others, regeneration may be incomplete or result in asymmetric limbs. This variability highlights the adaptability of echinoderms to their environment, where survival often necessitates the ability to recover from injury. The arm regrowth also involves the re-establishment of essential systems, including muscles, nerves, and even the ability to regenerate the central disc if severed. This capability underscores the evolutionary success of sea stars, allowing them to thrive in diverse marine habitats. As studies continue, researchers aim to uncover the molecular mechanisms driving regeneration, shedding light on how these creatures maintain resilience in the face of adversity. In understanding these processes, potential applications in regenerative medicine for humans may emerge, offering insights into healing and recovery. The mechanism of autotomy in echinoderms reveals a fascinating adaptation that enhances their survival. These marine organisms, which include starfish and sea cucumbers, possess the remarkable ability to shed parts of their bodies in response to threats. This loss, while seemingly detrimental, serves as a strategic maneuver to evade predators. Following autotomy, the regeneration process begins. Specialized cells known as blastomeal cells migrate to the site of the injury. These cells are crucial in orchestrating the complex rebuilding of lost structures. They proliferate and differentiate into various cell types. In starfish, for instance, the regeneration of limbs can occur with astonishing speed. The new arm develops from the central disc, ensuring that the echinoderm can quickly restore its functionality. Sea cucumbers exhibit a similar process, regenerating lost body parts and even internal organs. The implications of these regenerative strategies extend beyond the ocean floor. Insights gained from studying echinoderms may influence biomedical research, particularly in understanding human tissue regeneration. The mechanisms that underpin these processes offer potential pathways for advancements in healing techniques and regenerative therapies. Flatworms epitomize remarkable regenerative capabilities, able to restore lost body parts with astonishing efficiency. When a flatworm is severed, it will regenerate both anterior and posterior segments, showcasing a remarkable ability to reconstruct its entire body from a small fragment. This intricate process begins with the formation of a structure known as the blastoma, a mass of cells capable of developing into various tissues. Undifferentiated cells, or neoblasts, play a pivotal role in this regeneration. These cells are distributed throughout the organism and can transform into any cell type needed for repair. As the flatworm heals, they migrate to the injury site, proliferating and differentiating to form the necessary tissues and structures. The presence of specific signaling pathways, including the Wnt and hedgehog pathways, directs the regeneration process. These pathways are integral in orchestrating cellular behavior, ensuring that the new tissues develop correctly and integrate seamlessly with existing structures. Research into the regenerative mechanisms of flatworms has implications for a broader understanding of regeneration across species. Insights gained from these organisms may inform strategies for regenerative medicine in humans, paving the way for innovative treatments for injuries and degenerative diseases. The simplicity and efficiency of flatworm regeneration continue to inspire scientific inquiry into the mysteries of biological repair. Crustaceans exhibit a remarkable capacity for regeneration, particularly evident in the case of the American lobster. This species possesses the ability to regenerate lost limbs and claws, a process that reflects its evolutionary adaptations to predation. Each year, these creatures may lose appendages due to encounters with predators or environmental hazards, yet they possess the biological tools necessary to restore themselves. The regeneration process begins with the formation of a blastema, a mass of cells capable of growth and regeneration. This cellular cluster differentiates into various tissues, eventually leading to the development of a new limb. The regeneration occurs in stages, with initial growth characterized by a soft, cartilaginous structure that gradually hardens into the familiar chitinous exoskeleton. This transformation is not instantaneous. Rather, it can take several molts before the new appendage fully resembles its original counterpart. Interestingly, the regeneration process is influenced by factors such as age and environmental conditions. Younger lobsters tend to exhibit more efficient regeneration compared to their older counterparts. It is a reflection of their metabolic rates and overall vitality. This phenomenon prompts further questions into the genetic and environmental factors that govern regenerative capabilities, underscoring the complexity of biological systems. In the depths of the ocean, these resilient creatures continue their life cycles, embodying the intricate balance of survival and adaptation. The sea cucumber exemplifies this remarkable ability to regenerate. With its soft, leathery body, it employs a unique strategy to recover from injury. When threatened, it can expel its internal organs, creating a distraction that allows it to escape. Remarkably, these organs can regenerate, a process that takes several weeks. This regeneration is facilitated by specialized cells that can differentiate into various tissue types, showcasing a level of cellular plasticity not commonly observed in other organisms. In its habitat, the sea cucumber plays a vital role in maintaining ecosystem health. By feeding on detritus and organic matter on the ocean floor, it helps recycle nutrients, contributing to the overall productivity of its environment. This nutrient cycling is crucial for sustaining the diverse life forms that inhabit these depths. The regenerative capabilities extend beyond mere survival tactics. Researchers study the molecular mechanisms involved, focusing on the signaling pathways that govern regeneration. Understanding these processes could have profound implications for regenerative medicine in humans, offering insights into healing injuries and tissue repair. In laboratories, scientists examine the genetic underpinnings that allow for such extensive regeneration. The findings may unlock secrets of cellular regeneration, paving the way for groundbreaking advancements in medical science. This investigation into the sea cucumber's biology continues to reveal the complexities of life in ocean ecosystems. Fish regeneration showcases another remarkable phenomenon, particularly through the study of zebrafish. This small freshwater species possesses an extraordinary ability to regenerate various organs and tissues, including fins, heart, and even parts of its central nervous system. Research has demonstrated that upon injury, specialized cells in zebrafish rapidly mobilize to the site of damage. These cells then undergo a transformation, proliferating and differentiating into the necessary tissues to restore function. The presence of a cellular structure known as a blastema plays a pivotal role in this regenerative process. This collection of undifferentiated cells acts as a reservoir, providing the building blocks required for tissue restoration. Unlike in many other species, these cells retain a remarkable plasticity, allowing them to adapt and form different types of tissues. The regenerative capability of zebrafish has attracted significant scientific interest, leading to deeper inquiries into the underlying genetic and molecular mechanisms. Additionally, the environmental conditions in which zebrafish thrive influence their regenerative ability. Factors such as water temperature, chemical composition, and the presence of specific growth factors can enhance or inhibit regeneration. Understanding these influences not only sheds light on the biology of zebrafish, but may also offer insights applicable to regenerative medicine in humans, potentially advancing therapies for injuries and degenerative diseases. Heart regeneration in zebrafish presents a remarkable case study in biological resilience. Following injury, these creatures exhibit an extraordinary ability to repair their cardiac tissue, a process that remains largely elusive in mammals. The intricate mechanisms underlying this regenerative capacity involve a complex interplay of cellular signaling pathways and gene expression profiles. After cardiac damage, stem cells migrate to the site of injury, differentiating into cardiomyocytes, the heart muscle cells essential for function. This regeneration process is not just a matter of stem cell proliferation. It includes the reorganization of existing tissue, which facilitates the restoration of structural integrity and functionality. The role of specific growth factors and hormones has been extensively studied, revealing that they guide the cellular response and promote tissue remodeling. Understanding these biological processes in zebrafish provides a model for potential therapeutic applications in humans. By deciphering the molecular signals that prompt heart regeneration, researchers can aim to replicate these mechanisms in human medicine. The implications extend beyond just cardiac injuries. Insights gained could revolutionize treatment strategies for various heart diseases. Ongoing studies are exploring how these principles can be harnessed, potentially transforming the landscape of regenerative therapies and improving outcomes for patients suffering from cardiac conditions. The epicardium plays a crucial role in cardiac regeneration, acting as a protective layer that envelops the heart. This thin membrane is not merely a passive structure, it is actively involved in the regenerative processes following injury. Upon damage, the epicardium can respond by undergoing a series of complex biological events that facilitate healing and tissue repair. Recent research indicates that the epicardium releases various signaling molecules which assist in recruiting other cell types to the site of injury. These signaling pathways are critical in orchestrating the repair process, allowing for the regeneration of cardiac tissue. The cells derived from the epicardium can differentiate into myocytes, contributing to the regeneration of the heart muscle itself. In this region, the interaction between the epicardium and the underlying myocardium is pivotal. This relationship influences the proliferation of cardiac stem cells and their subsequent differentiation into functional heart cells. Furthermore, the epicardium can modulate the inflammatory response, promoting a more favorable environment for recovery and regeneration. Understanding the mechanisms employed by the epicardium may lead to novel therapeutic strategies aimed at enhancing heart repair. This ongoing investigation provides insight into how the body's innate regenerative capabilities can be optimized, holding promise for future advancements in treating heart disease and improving patient outcomes. Evolutionary perspectives illuminate the varied regenerative capabilities observed across vertebrate species. These adaptations have emerged through millions of years of natural selection, allowing certain organisms to recover from injuries that would be irreversible for others. The presence of regeneration in axolotls, for instance, reveals a sophisticated biological framework, enabling them to regenerate limbs, spinal cord, and even parts of their heart. This remarkable ability suggests an evolutionary advantage, allowing them to thrive in environments where predation is a constant threat. Comparative studies highlight the divergence in regenerative potential among different vertebrates. While some species exhibit limited regenerative properties, others, like zebrafish, can regenerate fins and parts of their heart with remarkable efficiency. This variability raises questions about the underlying genetic mechanisms that govern these processes. Investigating the molecular pathways involved reveals a complex interplay of growth factors and gene expression that varies significantly across species. Research into the evolutionary history of these abilities continues to uncover insights into the ancestral traits shared by vertebrates. Fossil evidence indicates that some regenerative features may have been present in early vertebrates, suggesting a long-standing relationship between regeneration and survival in changing environments. Understanding these evolutionary dynamics provides a holistic perspective on the regeneration phenomenon, guiding future research toward unlocking similar capabilities in mammals and other species. Regenerative strategies in vertebrates exhibit remarkable diversity across various species. In this realm, salamanders stand out, possessing the extraordinary ability to regenerate limbs, tails, and even parts of their heart and eyes. This region of study reveals that, unlike mammals, which typically rely on scar tissue formation following injury, amphibians utilize a process known as dedifferentiation. Cells at the injury site revert to a more primitive state, allowing them to proliferate and differentiate into the specific tissues required for regeneration. Fish also display significant regenerative abilities. Zebrafish, for instance, can regenerate fins and even heart tissue with relative ease. Investigations into their regenerative mechanisms reveal that they employ a combination of cellular proliferation and migration, enabling the rapid restoration of damaged structures. The presence of a regenerative blasema, a mass of cells capable of growth, is a critical feature in both zebrafish and salamanders. In contrast, reptiles exhibit a different approach, primarily focusing on tail regeneration. The mechanisms involved often include a complex interplay of growth factors and signaling pathways. Lizards can regrow their tails through a process that involves the formation of a cartilage rod instead of bone, highlighting the evolutionary adaptations to their specific ecological niches. These diverse strategies underscore the evolutionary innovation inherent in vertebrates, each adapting uniquely to their environments, while providing invaluable insights into regenerative biology. Tail loss serves as a defense mechanism in many reptiles, allowing them to escape predation. This remarkable ability is not merely a random occurrence, but a finely tuned survival strategy observed in species such as skinks and antelopes. The process begins with a well-defined physiological response, triggering the release of various signaling molecules that orchestrate cellular behavior at the injury site. The initial phase of regeneration involves the formation of a blastema, a mass of undifferentiated cells that serve as a foundation for new tissue development. Gradually, these cells differentiate into the various components needed for a functional tail, including muscle, cartilage, and skin. This regenerative process, while efficient, does not produce an exact replica of the original structure. Instead, the regrown tail displays distinct differences, such as the absence of vertebrae, replaced by a rod of cartilage. Research into these phenomena has broadened the understanding of cellular plasticity and the genetic mechanisms underlying regeneration. Insights gleaned from reptiles have sparked interest in potential applications for human medicine, particularly in developing therapies for tissue repair and regeneration. The exploration of these regenerative capabilities continues to unfold, revealing the intricate biological pathways that enable some reptiles to thrive despite significant physical loss. Lizard tail regeneration exemplifies complex biological mechanisms at work. Following the loss of a tail, a cascade of cellular events is triggered. This begins with the formation of a blastema, a dense cluster of cells that serve as the foundation for new tissue. Within this cluster, stem cells play a pivotal role, differentiating into various cell types required for tail reconstruction. The regeneration process also involves the reorganization of muscle, cartilage, and nerve tissue. Unlike mammals, which possess limited regenerative capabilities, lizards can restore not only the external structure but also internal components. Molecular signals guide cellular behavior, ensuring that the new tail maintains functional integrity. Regeneration is not merely a replacement of lost tissue. It is an intricate process that mirrors embryonic development. Cellular communication and signaling pathways are activated to orchestrate the growth. Factors such as growth factors and transcription factors are key players, directing the formation of new blood vessels and the re-establishment of nervous connections. Interestingly, the regenerated tail differs from the original in composition. The new structure often lacks vertebrae and instead consists of a cartilage rod. This adaptation may represent an evolutionary advantage, allowing for quicker recovery and survival in the wild. Understanding these mechanisms opens pathways for potential breakthroughs in regenerative medicine for humans, as scientists seek to harness nature's wisdom. Insights gleaned from avian regeneration reveal remarkable adaptations in feather and beak regrowth. The unique structure of feathers, composed of a protein called keratin, allows for a fascinating renewal process. Molting, a natural shedding and replacement cycle, occurs annually, ensuring that feathers remain functional and aerodynamic. During this cycle, weakened or damaged feathers are expelled, making way for new growth, which is initiated by complex hormonal signals. The regeneration of beaks presents an equally intricate process. Birds exhibit a remarkable ability to heal and reshape their beaks following injury. This occurs through the activation of specialized stem cells, which migrate to the damaged area and differentiate into various cell types necessary for rebuilding the beak structure. The process is influenced by environmental factors, nutrition, and the bird's overall health, emphasizing the interconnectedness of biological systems. Both feather and beak regeneration serve critical roles in avian survival. Feathers provide insulation, waterproofing, and the ability to fly, while beaks are essential for feeding and foraging. Any disruption in these regenerative processes can have significant consequences for a bird's survival. Studying these phenomena in birds contributes to a broader understanding of regenerative biology, offering potential strategies for enhancing healing mechanisms in other species, including humans. Epigenetics plays a crucial role in the regenerative processes observed in various organisms. This field examines how environmental factors can influence gene expression without altering the underlying DNA sequence. It provides insight into how certain species exhibit remarkable regenerative capabilities, allowing them to restore lost limbs or organs efficiently. In specific amphibians, such as salamanders, the ability to regenerate is linked to epigenetic modifications that activate specific genes essential for tissue growth and repair. These modifications can be triggered by physical stimuli or changes in the surrounding environment. The dynamics of these epigenetic changes are complex, often involving methylation patterns and histone alterations that regulate gene activity. By understanding these mechanisms, researchers aim to unlock the potential of epigenetic interventions in regenerative medicine. The implications extend beyond simple regenerations. They encompass the broader field of tissue engineering and regenerative therapies. Insights gained from studying these processes could aid in developing strategies for wound healing, organ repair, and even the reversal of degenerative diseases. The interplay between genetics and epigenetics highlights the intricate balance that governs regeneration, revealing a sophisticated network of biological interactions. This understanding of regenerative capabilities in nature provides a foundation for innovative approaches to enhance healing in various species, including humans. Environmental factors play a crucial role in the regenerative processes observed in various species. Conditions such as temperature, humidity, and the presence of certain chemicals can significantly influence the rate and efficiency of regeneration. For instance, warmer temperatures often accelerate metabolic processes, enhancing the healing capabilities of certain organisms. These changes can lead to more rapid cell division and tissue formation. Soil composition and water quality are similarly pivotal. Species inhabiting nutrient-rich environments may exhibit heightened regenerative abilities due to the availability of essential minerals and organic compounds. This relationship underscores the importance of habitat quality in fostering regeneration. Conversely, pollution or habitat degradation can impede these processes, leading to diminished healing capabilities over time. Light exposure also has a notable impact. Certain organisms, such as planarians, rely on specific wavelengths of light to regulate their regenerative cycles. The absence of adequate light can disrupt these biological rhythms, resulting in slower regeneration or incomplete healing. Seasonal variations further affect regeneration, with many species exhibiting heightened regenerative capabilities during specific times of the year. This cyclical behavior is often linked to reproductive cycles and environmental cues that signal optimal conditions for growth and recovery. Understanding these environmental influences enriches the knowledge of regenerative biology, paving the way for potential applications in medical science and conservation efforts. The intricate relationship between surroundings and regeneration remains a vital area of exploration. Nutritional factors significantly influence regenerative processes across various species. In environments rich in specific nutrients, organisms often exhibit enhanced capacity for regrowth. For instance, the availability of particular amino acids and vitamins can accelerate the healing of wounds and the restoration of lost limbs. This region, where food sources are plentiful, showcases remarkable regenerative feats, illustrating the profound connection between diet and biological renewal. Certain aquatic species, such as planarians, demonstrate this relationship vividly. When subjected to nutrient-rich conditions, these flatworms can regenerate their entire bodies from mere fragments. The mechanisms behind such growth involve complex biochemical pathways which are activated in response to abundant food supplies. It is in these environments that the effects of nutrition become most evident, driving cellular division and differentiation. Terrestrial species also exhibit similar dependencies. Salamanders, known for their regenerative abilities, rely on a diverse diet to replenish the essential nutrients necessary for limb regrowth. Without access to key nutrients, these processes are hindered, resulting in slowed regeneration or incomplete recovery. Understanding the precise nutritional needs required for optimal regeneration could pave the way for advancements in regenerative medicine. This knowledge holds potential not only for wildlife rehabilitation, but also for human applications, where nutritional interventions may enhance healing outcomes and recovery processes. The quiet influence of nutrition remains a fundamental aspect of regenerative biology. Molecular pathways involved in regeneration reveal intricate networks of signaling that govern cellular processes. These pathways are often activated in response to injury, initiating a cascade of biological events essential for tissue repair and growth. Factors such as growth factors, cytokines, and extracellular matrix components play crucial roles in this orchestration. In various species, particularly those with remarkable regenerative capabilities. Specific genes are upregulated, facilitating the transition from a wounded state to a regenerative one. The activation of pathways, such as the WNT and hedgehog signaling cascades, promote cellular proliferation and differentiation. These mechanisms enable cells to assume versatile roles, adapting to the requirements of the regenerating tissue. Regeneration often involves a unique reprogramming of somatic cells, allowing them to adopt stem cell-like properties. This cellular plasticity is critical as it provides the necessary resources for rebuilding lost or damaged structures. In some instances, the differentiation occurs where specialized cells revert to a more primitive state, capable of extensive division and specialization. Research continues to uncover how these molecular pathways interact and coordinate to achieve successful regeneration. The elucidation of these processes not only deepens the understanding of biological resilience, but also holds potential implications for regenerative medicine in humans, offering insights into healing and repair mechanisms that remain largely untapped. Hormones play a crucial role in determining the regenerative capacity of various species. These biochemical messengers regulate a multitude of physiological processes, influencing how organisms respond to injury and initiate repair. In certain amphibians, such as salamanders, the presence of specific hormones can trigger the regeneration of limbs and tails. This phenomenon is closely linked to the activity of growth factors and signaling pathways, which guide cellular proliferation and differentiation. In mammals, the influence of hormones on regeneration is more complex. While some hormones promote healing, others may inhibit it. For instance, the hormone cortisol, known for its role in stress responses, can impede the healing process by suppressing inflammation and altering immune functions. Understanding these dynamics is essential for harnessing the potential of regenerative therapies. Research into hormonal influence extends to the study of stem cells, which are pivotal in regeneration. Hormones such as insulin-like growth factor stimulate stem cell activity, enhancing their ability to regenerate tissue. This area of study holds promise for advancing treatments for degenerative diseases and injuries in humans. The intricate balance of hormone levels and their interactions with cellular processes underscores the complexity of regeneration. Insights gained from these investigations not only illuminate the regenerative capabilities of various species, but also pave the way for innovative medical approaches to enhance healing in humans. This region of study reveals the remarkable case of the African spiny mouse, found predominantly in arid environments. These small mammals possess an extraordinary ability to regenerate skin and fur. When subjected to injury, they can regrow entire patches of skin, complete with hair follicles, hair, and even sweat glands. This phenomenon is striking, particularly when compared with other mammals that exhibit limited regenerative capabilities. The mechanisms behind this regenerative prowess involve a complex interplay of cellular processes. Upon injury, specialized cells proliferate and migrate to the wound site, where they begin to rebuild the damaged tissue. This regeneration is not merely superficial. The new skin is fully functional, allowing the mouse to maintain its ecological adaptations in harsh surroundings. Research indicates that the African spiny mouse utilizes a unique strategy, including a reduced inflammatory response and enhanced cellular plasticity. These adaptations facilitate the rapid healing process, minimizing the risk of infections that often complicate recovery in other species. The implications of this research extend beyond understanding the biology of these resilient rodents. Insights gleaned from their regenerative capabilities may inform novel therapeutic strategies for treating wounds and injuries in humans. In examining the African spiny mouse of Courreau's pomme, the interplay between evolution and regeneration becomes increasingly clear, showcasing the potential for nature to inspire advancements in medical science. Hair regeneration in mammals presents a fascinating avenue for exploration, particularly in the context of the African spiny mouse. This species exhibits an extraordinary ability to shed its fur under duress, a process that is not merely a survival mechanism, but also a precursor to regeneration. The regrowth of hair occurs rapidly, demonstrating an efficient response to injury. Research indicates that specific skin cells in this region can transition between states, transitioning from a quiescent to an active phase during the regeneration process. The mechanisms behind this transformation reveal intricate signaling pathways that govern cellular behavior. Understanding these signals can elucidate how hair follicles can be reactivated and regenerated, a process that remains elusive in many other mammals. The role of stem cells in the regeneration of hair follicles is particularly noteworthy. These versatile cells possess the ability to differentiate into various cell types, contributing to the formation of new hair shafts. This phenomenon serves as a reminder of the untapped potential for therapeutic applications in hair loss conditions affecting humans. Investigations into the molecular biology of hair regeneration continue to uncover the genetic and environmental factors influencing this remarkable capability. Insights derived from the African spiny mouse may illuminate the potential for human regenerative capabilities. Research indicates that certain cellular mechanisms present in these mice offer a glimpse into how regeneration may be harnessed in humans. This region of study encompasses the exploration of stem cells, which play a pivotal role in tissue repair and regeneration across various species. In humans, the natural regenerative process, though limited, still exhibits remarkable features. For instance, the liver possesses a unique ability to regenerate after partial removal, capable of returning to its original size and functionality. This phenomenon reflects a complex interplay of cellular signaling and growth factors that orchestrate healing. Comparative analysis extends to species such as axolotls, which regenerate limbs, spinal cords, and even parts of their hearts. Insights from these organisms reveal that reprogramming cellular responses may hold the key to unlocking similar capabilities in humans. The presence of specialized cells, such as progenitor cells, supports this regenerative potential, though the mechanisms remain less efficient in human biology. Current research endeavors seek to bridge the gap between these regenerative marvels and human capacity. Understanding the intricate molecular pathways involved may pave the way for innovative therapies aimed at enhancing healing and regeneration in human tissues, emphasizing the evolutionary nuances of regenerative biology. The molecular pathways of regeneration in various species highlight significant limitations in human healing processes. While some organisms exhibit remarkable capabilities, such as regrowing limbs or organs, humans are bound by evolutionary constraints that dictate a more conservative approach to tissue repair. The process of wound healing in humans is primarily focused on restoring skin integrity, often leading to scar formation rather than regeneration of the original tissue. This region of biology reveals that the cellular mechanisms responsible for regeneration vary greatly among species. For instance, the ability of certain amphibians to regenerate limbs involves complex cellular reprogramming and de-differentiation, allowing specialized cells to revert to a more primitive state. Such processes remain largely inaccessible to human cells, which possess a limited capacity for plasticity. In humans, healing is typically characterized by inflammation, followed by tissue repair that does not fully restore the original architecture or function, Factors such as age, genetics, and environmental influences further complicate these processes, leading to variations in healing efficiency. The immune response of fulfill these processes, leading to variations in healing efficiency. The immune response of fulfill, while essential for defense, can impede regeneration, creating a delicate balance between healing and potential for restoration. Investigating the reasons behind these differences not only enhances our understanding of human biology, but also informs future research into regenerative medicine, aiming to bridge the gap between human healing and the extraordinary capabilities observed in nature. Scar formation represents a fundamental aspect of the healing process, often marking the transition from injury to recovery. The intricate mechanisms involved in this process highlight the complexities of tissue repair. In many species, healing results in the development of fibrous tissue, which replaces the original structure and function of the damaged area. This fibrotic response can restore integrity but may lack the precision that regeneration achieves. And in contrast, regeneration allows for the complete restoration of lost or damaged tissues, often without the formation of scars. This phenomenon is observed in species such as salamanders, which can regenerate limbs and other body parts with remarkable fidelity. The fundamental difference lies in the cellular response to injury. Regenerative species activate specific pathways that enable the reorganization of cells into their original forms, while healing in mammals often leads to the accumulation of collagen and other extracellular matrix components that form scar tissue. Understanding the molecular signals that govern these processes can illuminate potential pathways for improving human healing. By studying the regenerative capabilities of certain animals, researchers are identifying key factors that could be manipulated to enhance healing in humans. This research encompasses a myriad of cellular interactions, gene expressions, and environmental influences that govern the outcomes of tissue repair and regeneration. Advances in regenerative medicine have begun to illuminate the pathways of healing through stem cell therapy. This innovative approach harnesses the unique capabilities of stem cells, which possess the remarkable potential to differentiate into various cell types by isolating these cells from diverse sources, such as bone marrow or induced pluripotent stem cells. Researchers can create tailored treatments aimed at repairing damaged tissues and organs. Applications of stem cell therapy span a wide range of medical conditions, from degenerative diseases to traumatic injuries. In this context, the ability of stem cells to migrate to areas of injury and promote repair through paracrine signaling is of particular interest. It fosters an environment conducive to healing, enabling the regeneration of tissues that were once thought irreparable. Studies continue to refine the techniques involved in stem cell manipulation and delivery, optimizing their effectiveness in clinical settings. The challenge of understanding the immunological responses elicited by these cells remains a focal point for researchers. Precision in targeting specific tissues while minimizing adverse effects is crucial. These developments not only offer hope for improved therapeutic outcomes, but they also pave the way for a deeper understanding of the fundamental biological processes that govern regeneration. Continued exploration in this field holds promise for transforming the landscape of medicine. Bioengineering emerges as a vital field, leveraging insights from regenerative biology to enhance tissue regeneration. Researchers are employing advanced materials, such as hydrogels and biocompatible scaffolds, to create environments that mimic natural tissue structures. These innovations facilitate cellular growth and organization, fostering the regeneration of complex tissues. Among the most promising trends, three-dimensional bioprinting stands out, enabling the precise arrangement of cells and materials in layers. This method holds the potential to produce organ-like structures, paving the way for future transplant solutions. The advent of stem cell technology further amplifies progress in this realm. Stem cells possess the unique ability to differentiate into various cell types, making them invaluable for regenerating damaged tissues. Clinical trials are ongoing, exploring their application in treating conditions such as spinal cord injuries and heart disease. Scientists are also investigating the genetic pathways that govern regeneration, seeking to unlock the mechanisms that allow certain species to regrow lost limbs or organs. Collaboration between disciplines enhances these efforts, combining insights from biology, engineering, and materials science. As knowledge expands, so does the potential for bioengineered solutions to address pressing medical challenges. The exploration of these avenues continues to spark innovation, bringing humanity closer to harnessing the power of regeneration for therapeutic applications. Ethical considerations in regenerative research weigh heavily on the minds of scientists and ethicists alike. The potential for significant advancements must be balanced with the implications of manipulating biological systems. Questions arise regarding the extent to which it is acceptable to alter the natural processes of life. The responsible pursuit of knowledge involves scrutinizing the possible outcomes of such interventions. In this context, the treatment of animal models plays a critical role. The welfare of these subjects must be prioritized, ensuring that research adheres to humane principles. The necessity of obtaining informed consent becomes complex when considering non-human entities. A careful evaluation of the moral responsibilities towards these beings informs the ethical landscape of regenerative studies. Long-term consequences of regenerative techniques are also essential to consider. The introduction of modified organisms into ecosystems may lead to unforeseen disruptions. This region of inquiry emphasizes the importance of rigorous oversight and thorough assessments prior to implementation. The dialogue surrounding regenerative research is ongoing, marked by diverse perspectives. Stakeholders from multiple disciplines contribute to this evolving conversation. The challenge lies in navigating the intricate balance between scientific advancement and ethical integrity. This commitment to thoughtful exploration will shape the future of regenerative medicine while safeguarding the values that underpin responsible research. The role of genetics in regenerative abilities is fundamental, guiding the processes through which organisms repair and regenerate tissues. Specific genes dictate the pathways for cell differentiation and proliferation, influencing the extent to which regeneration can occur. In certain species, a unique set of genes orchestrates the regeneration of limbs or organs, revealing a sophisticated molecular framework that governs these remarkable capabilities. Research into these genetic mechanisms has unveiled that regenerative potential is often tied to the expression of particular genes during critical developmental stages. In some cases, the ability to regrow limbs can be traced back to embryonic development, wherein these organisms retain cellular plasticity long after maturation. The manipulation of these genetic pathways could advance therapeutic techniques for humans, offering insights into potential treatments for injuries and degenerative diseases. Molecular signals that facilitate regeneration have been identified, highlighting the intricate communication between cells. Factors such as growth hormones and signaling proteins play pivotal roles in initiating and sustaining the regenerative processes. The study of organisms with extraordinary regenerative capabilities, such as axolotls and certain species of salamanders, continues to inform our understanding of these genetic networks. Unlocking the secrets of regeneration through genetics not only broadens the horizons of biological science, but also offers practical applications that could transform medical practices and enhance healing methodologies in the future. The mechanisms that facilitate regeneration are intricately linked to the biological processes of aging. Over time, cellular functions decline, impacting the efficiency with which organisms can repair and regenerate tissues. In many species, The regenerative capabilities observed in youth diminish significantly as they age. This phenomenon is particularly evident in mammals, where the ability to heal from injuries or diseases becomes markedly less effective with advancing age. Research into this aspect unveils a complex interplay of genetic, metabolic, and environmental factors. Some organisms exhibit remarkable longevity and retain their regenerative prowess well into adulthood. The study of these exceptions sheds light on the potential for enhancing regenerative abilities in other species, including humans. Specific genes and cellular pathways involved in regeneration may hold the key to mitigating the effects of aging. In certain marine species, such as starfish and some types of fish, the ability to regenerate limbs and other body parts persists throughout their lifespan. Understanding the genetic foundations that allow these organisms to maintain their regenerative capabilities could lead to significant breakthroughs in regenerative medicine. Insights gained from these studies may pave the way for therapies aimed at counteracting age-related decline, potentially unlocking new avenues for treating degenerative conditions and enhancing overall health during the later stages of life. Regeneration encompasses the ability to completely restore lost or damaged structures, often resulting in full functional recovery. In contrast, repair refers to the process of healing that may not fully restore the original form or function of an organism. This distinction is critical in understanding the biological mechanisms underlying various healing processes. Certain species, such as salamanders and axolotls, exhibit remarkable regenerative capabilities. They can regrow limbs, tails, and even sections of their hearts and spinal cords. The complex interplay of cellular signaling, gene expression, and stem cell activity plays a pivotal role in this regeneration. These mechanisms provide insights into how organisms manage to re-establish intricate anatomical structures. Repair mechanisms, on the other hand, tend to involve the formation of scar tissue, which serves to close wounds and restore integrity, but may not restore full functionality. In mammals, for instance, heart tissue damaged by a myocardial infarction often heals through repair, leading to scar formation rather than regeneration. This results in altered function and potential long-term complications. Comparing these two processes reveals not only the evolutionary adaptations of various species, but also highlights the potential for advancing medical science. Understanding the underlying principles of regeneration could inspire novel therapeutic techniques, enabling improved healing and restoration in organisms that currently rely on repair mechanisms alone. The immune response plays a crucial role in the regeneration process, acting as both a facilitator and a regulator. This complex system responds to injury by mobilizing various cell types and signaling molecules, orchestrating healing processes that initiate tissue repair. In many organisms, the initial stages of regeneration are marked by inflammation, which serves to clear debris and prevent infection. These immune cells can release growth factors that promote cellular proliferation and migration to the site of injury. In certain species, such as axolotls and planarians, the immune cells are not merely passive responders, but actively contribute to the regeneration process. Their presence can influence the formation of new tissues, helping to guide stem cells toward differentiating into the appropriate cell types required for regeneration. For instance, specific immune cells have been observed to secrete factors that encourage the growth of muscle, nerve, and skin tissues. The relationship between the immune system and regenerative capacity suggests that evolution may have fine-tuned these responses over time. Organisms capable of extensive regeneration have developed unique immune profiles that support their regenerative abilities. Conversely, in species with limited regenerative potential, chronic inflammation can hinder healing, reflecting a balance that is essential for optimal recovery. Understanding these interactions could provide insights into enhancing regenerative therapies in humans. The intricate relationships between organisms and their microbiomes play a crucial role in regenerative processes. Microbiomes, communities of microorganisms residing within and on hosts, influence a variety of biological functions, including immune responses and metabolic pathways, in organisms capable of remarkable regeneration, such as axolotls and certain species of starfish. These microbial communities contribute significantly to tissue repair and regeneration. Research indicates that specific microbial populations can enhance the regenerative capabilities of these animals. In some cases, the presence of particular bacteria helps modulate inflammatory responses, creating an environment conducive to healing. This region's microbial diversity appears to facilitate the necessary biochemical processes that underpin regeneration, suggesting a symbiotic relationship that promotes recovery from injury. Observations indicate that disruptions in microbial communities can impair regeneration. For instance, in studies involving the removal of certain bacteria, researchers noted a marked decline in the regenerative efficiency of model organisms. This correlation highlights the potential for microbiome manipulation as a strategy to improve regenerative outcomes. Current efforts focus on understanding the precise mechanisms through which these microorganisms operate. By identifying key bacterial species and their functions, scientists aim to develop targeted therapies that mimic or enhance the beneficial effects of microbiomes. This exploration could pave the way for innovative approaches to regenerative medicine in humans, unlocking new avenues for healing and restoration. Cross-species regeneration presents a fascinating study of nature's remarkable capabilities. Various organisms demonstrate extraordinary abilities to regenerate lost structures, offering insight into cellular mechanisms that remain enigmatic. In this region, the axolotl stands out, capable of regrowing limbs, spinal cord, heart tissue, and even parts of its brain with remarkable precision. This creature relies on a unique population of stem cells that remain undifferentiated, allowing them to transform into necessary cell types during the healing process. Other inhabitants of the animal kingdom exhibit different regenerative strategies. The planarian, for example, can regenerate its entire body from a small fragment. This efficiency stems from a high concentration of pluripotent stem cells, which are instrumental in restoring functionality. The simplicity of its body plan contrasts sharply with the complexity of higher organisms, yet the underlying principles of regeneration offer profound implications for understanding cellular behavior. Sea cucumbers utilize a distinct approach, expelling their internal organs as a defense mechanism and later regenerating them. This process reflects an adaptive strategy to survival. Observations of these diverse methods reveal that regeneration is not merely a biological curiosity, but an evolutionary response, shaped by environmental demands and challenges. The implications of these findings resonate of Mieldth, suggesting pathways to enhance regenerative capabilities within more complex systems. Biomimicry provides a fascinating framework for understanding and applying these regenerative techniques observed in nature. This domain of study examines how organisms, from the humble axolotl to the resilient planarian, employ intricate biological processes for regeneration. Researchers analyze the underlying mechanisms, identifying genetic and cellular pathways that facilitate such remarkable feats. These insights serve as inspiration for innovative materials and solutions in various fields. For example, the structural properties of spider silk are being emulated to create stronger and lighter synthetic fibers. Similarly, the regenerative capabilities of certain marine organisms inform advancements in wound healing technologies. Nature's methods offer lessons in efficiency and sustainability, emphasizing the importance of adaptive responses to environmental stresses. By mimicking these biological processes, scientists aim to design systems that can self-repair, reducing waste and extending the lifespan of materials. Cultivating a deeper understanding of how these organisms thrive in their environments allows for the development of bio-inspired technologies. Research continues to uncover the genes responsible for these regenerative processes, potentially unlocking new medical treatments for humans. As this field evolves, collaborative efforts between biologists, engineers, and material scientists pave the way for transformative solutions grounded in the wisdom of nature's designs. The quest to harness these principles reflects a profound appreciation for the intricate balance of life. Regenerative medicine stands at the threshold of remarkable advancements, driven by an understanding of these biological phenomena. Potential applications extend across various fields, offering solutions that could redefine medical practice. Tissue engineering emerges as a pivotal area, where scientists seek to create synthetic organs and tissues that mimic natural structures. By utilizing stem cells and biomaterials, they aim to address the chronic shortage of donor organs. Wound healing represents another promising frontier. Enhanced regenerative therapies could significantly improve recovery times, allowing for faster healing of severe injuries. This region of research focuses on developing bioactive dressings that release growth factors, directly stimulating tissue regeneration. In orthopedics, the ability to regenerate cartilage and bone could revolutionize treatment for degenerative diseases and traumatic injuries alike. The field of neurology also stands to benefit, particularly through the regeneration of nerve cells. Breakthroughs in this area could lead to effective therapies for spinal cord injuries and neurodegenerative diseases, restoring function and improving quality of life for countless individuals. Beyond physical healing, the psychological impact of regenerative medicine cannot be understated, as these advancements offer hope to patients grappling with the limitations imposed by injury and disease. As exploration continues, ethical considerations will inevitably arise, shaping the future landscape of regenerative solutions. Scientists and ethicists must navigate these complexities carefully, ensuring responsible progress. The quest for bio-artificial organs represents a significant frontier in regenerative medicine. This field seeks to replicate the remarkable ability of certain species to regenerate lost tissues and organs. Research has increasingly focused on understanding the mechanisms underlying these natural processes, aiming to apply this knowledge to human health. Advances in biotechnology and tissue engineering have led to the development of scaffolds that mimic the extracellular matrix, providing a supportive structure for cell growth. The integration of stem cell technology plays a crucial role in this endeavor. Stem cells possess the ability to differentiate into various cell types, offering the potential to generate functional tissues. This region of research encompasses not just organ replacement, but also the creation of lab-grown tissues that can be used for drug testing and disease modeling. Initial experiments have demonstrated the feasibility of constructing rudimentary organs, such as bladders and hearts, in controlled laboratory settings. Challenges remain, particularly in vascularization, the complex network of blood vessels required to sustain larger or more complex organs. Researchers are employing innovative methods, including three-dimensional bioprinting, to address this issue. Each advancement brings scientists closer to a future where organ transplantation may no longer be dependent on donor availability, fundamentally transforming the landscape of medicine and patient care. This transformation in medicine is being bolstered by innovations in three-dimensional bioprinting. This technology enables the precise layering of cells and biomaterials, creating living tissues that can mimic the structure and function of natural organs. The bioprinting process involves a computer-aided design, allowing for detailed customization to meet specific patient needs, thereby enhancing the potential for personalized medicine. Researchers are exploring various applications of this technology, particularly for skin, cartilage, and vascular tissues. In burn treatment, for instance, bioprinted skin can provide immediate coverage, promoting healing and reducing the risk of infection. Cartilage regeneration is being investigated for applications in joint repair, offering hope to those suffering from degenerative diseases. Our vascular tissues hold significant promise as well, as they can facilitate the integration of bioprinted structures with the host's circulatory system, supplying necessary nutrients and oxygen. This integration is crucial for the viability of larger tissue constructs, which require a stable blood supply. As the science of three-dimensional bioprinting evolves, it is poised to address the challenges associated with organ shortages. Continued advancements could lead to the eventual creation of fully functional organs, offering a potential solution to the limitations of traditional organ transplantation. This future may redefine the parameters of regenerative medicine, emphasizing the importance of innovation and research in enhancing human health. Gene editing plays a pivotal role in enhancing the regenerative capabilities observed in certain animals. Techniques such as CRISPR-Cas9 allow for precise modifications of genetic material, unlocking pathways that enable regeneration. These innovations provide insight into the fundamental mechanisms behind cellular repair and regeneration. In this context, various species exhibit remarkable feats of regeneration, such as the axolotl, which can regrow entire limbs. It possesses specialized cells that can convert into various cell types during the regeneration process. Researchers are investigating the genetic basis of this ability, identifying genes responsible for cellular plasticity and growth. The potential applications of these discoveries extend beyond mere understanding. By manipulating specific genes, scientists aim to induce similar regenerative processes in mammals, including humans. The implications could transform approaches to treating injuries and degenerative diseases. Gene editing not only reveals the latent regenerative pathways, but also presents ethical considerations. The balance between advancement and caution becomes crucial as capabilities grow. Ensuring responsible application of these technologies remains a vital discussion among scientists and ethicists alike. Advancements in gene editing illustrate a frontier where biology and technology converge. The ongoing research promises to unveil the complexities of regeneration, paving the way for groundbreaking therapies that could one day repair damaged tissues and restore functionality. Overcoming barriers in regenerative medicine presents a multifaceted challenge. One significant hurdle lies in understanding the mechanisms that underlie the regenerative capabilities observed in certain species. This region of study demands an intricate examination of cellular processes, signaling pathways, and gene expression patterns. Identifying how these processes can be replicated in humans remains a formidable task. In addition, the immune response complicates efforts to harness regenerative techniques. The body's defense mechanisms often target and reject transplanted tissues or engineered cells, impeding successful integration. This phenomenon necessitates the development of advanced materials and methods to promote acceptance and minimize rejection. Another challenge stems from the complexity of human anatomy and the variability among individuals. Regenerative therapies must account for distinct genetic backgrounds, environmental factors, and pre-existing conditions that influence healing. This variability complicates the standardization of treatments, demanding a more personalized approach to regenerative medicine. Ethical considerations also emerge, particularly regarding the manipulation of stem cells and genetic engineering. These ethical dilemmas require careful contemplation, balancing potential benefits against moral implications. With ongoing advancements in technology and a deeper understanding of biology, pathways to overcome these obstacles are gradually being illuminated. Each step forward in this intricate landscape brings with it the promise of transforming medicine and enhancing the quality of life for countless individuals. Successful regenerative treatments serve as compelling evidence of the potential inherent in biological systems. One prominent example involves the use of stem cells derived from various tissues to repair damaged organs. In clinical settings, these cells have demonstrated the ability to regenerate cardiac tissue following myocardial infarctions, leading to improved heart function in patients who once faced grim prognoses. This region of research continues to expand as scientists explore variations in stem cell applications across different organ systems. Another notable case involves the therapeutic use of biomaterials designed to support tissue regeneration. In orthopedic medicine, scaffolding made from biodegradable materials has facilitated the healing of bone fractures that were previously slow to recover. The integration of these scaffolds with the body's natural healing processes has led to enhanced recovery times and restored mobility for many individuals. Flow-efficient on Surian represents a further advancement where laboratory-grown organs are developed for transplantation. Recently, successful trials have been conducted with engineered skin capable of restoring protective barriers for burn victims. These innovations provide insights into how regenerative capabilities can be harnessed, potentially addressing shortages in donor organs. This growing body of research underscores the multifaceted approaches being employed in regenerative medicine, revealing a landscape rich with possibilities that may redefine therapeutic interventions in the future. The interplay of biophysical forces within regenerative processes reveals critical mechanisms that influence outcomes. Mechanical forces, such as tension and compression, shape tissue architecture and cellular behavior. These forces can dictate the alignment of cells, guiding them towards organized structures essential for functional regeneration. Cellular responses to these forces include changes in gene expression, altering pathways that govern growth and differentiation. The extracellular matrix serves as a dynamic scaffold, responding to mechanical stimuli by facilitating communication between cells and their environment. As a result, the matrix not only provides structural support, but also actively participates in directing regeneration. Fluid movement and shear stress contribute significantly to cellular signaling during regenerations. In aquatic environments, for instance, the flow of water can enhance nutrient distribution, enabling cells to proliferate more effectively. This mechanism is particularly evident in organisms that exhibit remarkable regenerative abilities, such as certain species of salamanders and planarians. Understanding these biophysical forces offers a pathway to enhance regenerative strategies in humans. Researchers are exploring how to manipulate mechanical environments in tissue engineering to promote healing and regeneration. By integrating these principles, the potential exists to create more effective therapies, transforming the landscape of regenerative medicine and its applications. The scientific community continues to explore these dimensions, seeking to unlock the full potential of regeneration. Neural regeneration represents a complex frontier within the study of cellular recovery. Unlike many organisms that exhibit remarkable regenerative capabilities, mammals possess limited abilities to repair and regenerate neural tissues. This restriction is particularly evident in the central nervous system, where injuries often lead to permanent damage. Research has illuminated various mechanisms that underlie this phenomenon, revealing the intricate interplay between cellular behaviors and environmental influences. In this region, glial cells play a pivotal role. These supportive cells respond to injury by proliferating and forming a scar. While this response may initially stabilize the area, endormert trophic factors emerge as key players in this process, promoting cell survival and growth. Investigations into their mechanisms may offer pathways to enhance regeneration in mammals. The exploration of these insights fosters hope for future treatments, not only for spinal cord injuries, but also for neurodegenerative diseases. Continued inquiry into neural regeneration stands at the intersection of biology and medicine, aiming to bridge the gaps in understanding and application. The nervous system plays a critical role in the process of limb regeneration, guiding the complex orchestration of cellular behaviors. In species capable of this remarkable feat, such as axolotls, the presence of nerve fibers near the amputation site initiates a cascade of biological events. These nerves release signaling molecules that stimulate surrounding cells, prompting them to enter a regenerative state. This region becomes a hub of activity as cells swiftly de-differentiate, losing their specialized functions to revert to a more primitive state. This reversion is essential, providing the necessary building blocks for the regeneration process. Stem cells proliferate and migrate towards the injury, forming a structure known as the blasema, which serves as the template for the new limb. Intricate communication occurs between the neural tissue and the regenerating cells. The signals from the nervous system not only promote cell division, but also help in establishing the correct spatial organization. This coordination is vital for ensuring that the regenerated limb possesses the correct anatomical structure and functionality. In addition to promoting growth, the nervous system also plays a role in the sensory feedback necessary for the developing limb. Sensory neurons provide crucial information, allowing the organism to adapt its behavior around the newly formed structure, ensuring survivability and proper integration into the environment. Regenerative evolution illustrates a complex interplay between adaptation and survival in various species. In environments marked by harsh conditions, organisms often develop remarkable regenerative traits to cope with loss or damage. These adaptations are not merely responses to injury, they represent a dynamic evolutionary process shaped by selective pressures over generations. While some species possess inherent capabilities to regenerate complex structures, others exhibit a more gradual enhancement of these traits through environmental interactions. In this region, certain amphibians display the ability to regrow limbs, a trait that evolves in response to predation and habitat destruction. The underlying genetic mechanisms reveal a sophisticated network of signaling pathways that facilitate regeneration. This process often requires the precise coordination of stem cells and growth factors, highlighting the intricate balance of biological systems. Observations in marine environments showcase the resilience of echinoderms, which can regenerate lost arms and even internal organs. Their evolutionary adaptations demonstrate a successful strategy for survival against natural threats. Such regenerative capabilities allow for a flexible approach to environmental challenges, ensuring that these organisms can thrive despite fluctuations in their habitats. Each instance of regeneration serves as a testament to the power of evolution, revealing not just the ability to restore lost parts, but also an opportunity to adapt and persist in an ever-changing world. Insights from regeneration illuminate pathways for understanding evolutionary biology. The mechanisms that underpin these remarkable abilities often reveal intricate genetic and molecular processes. Studies on species such as axolotls and certain species of starfish have unveiled the roles of specific genes in tissue regrowth and cellular reprogramming. This exploration provides a foundation for unraveling fundamental biological questions, including how complexity arises in multicellular organisms. The future of evolutionary biology may hinge on these findings, offering potent implications for medicine and biotechnology. By deciphering the regenerative codes embedded within these organisms, researchers could potentially harness similar mechanisms to promote healing in humans. The prospect of regenerating damaged organs or tissues parallels the natural processes observed in these resilient species. This approach invites a re-evaluation of evolutionary timelines, suggesting that the capacity for regeneration might not be as rare as previously thought. It raises questions about the evolutionary pressures that favor such traits, particularly in environments marked by instability. Over time, these insights could lead to a deeper understanding of the evolutionary landscape, where adaptability and resilience become key survival strategies. The implications extend beyond the laboratory, influencing conservation efforts and ecological studies. Understanding regeneration not only enriches the scientific narrative, but also highlights the interconnectedness of life within diverse ecosystems. Pathways to understanding regenerative failure reveal critical insights into biological limitations and the mechanistic barriers faced by various species. This realm of study examines the factors that inhibit the process of regeneration, often shedding light on evolutionary adaptations. In some instances, organisms possess regenerative capabilities that diminish with age or environmental stresses, leading to a decline in tissue repair and cellular renewal. Research indicates that cellular senescence plays a pivotal role, where damaged cells cease to divide and can disrupt the regenerative process. This phenomenon is notably observed in certain mammalian species, including humans, where the capacity for regeneration is significantly constrained. The accumulation of genetic mutations over time further compounds this issue, resulting in diminished cellular function and an inability to effectively regenerate lost or damaged tissues. Investigations into the signaling pathways involved in regeneration have identified key molecules that govern cellular responses. These molecular signals can either promote or inhibit regeneration, depending on the organism and the specific context. The study of regenerative failure not only illuminates the challenges faced by various species, but also informs regenerative medicine by highlighting potential targets for therapeutic intervention. This exploration of regenerative limitations, investigating the role of apoptosis in regenerative processes, reveals a complex interplay between cell death and tissue renewal. This phenomenon, often perceived merely as a destructive force, serves as a crucial component in the orchestration of regeneration. In many species, it is not the absence of apoptosis that leads to successful regeneration, but rather its precise timing and regulation. Cells destined to die through apoptosis often release signaling molecules that can stimulate neighboring cells, prompting them to enter a state of proliferation and differentiation. This regenerative response is observed in various organisms, from amphibians to certain species of fish, which can effectively replace lost or damaged structures. The mechanisms underlying this process are intricate. Specific pathways governing the apoptotic process influence how these organisms manage cellular turnover, while simultaneously fostering the regrowth of limbs or organs. Research into these pathways has uncovered potential targets for enhancing regenerative capacities in species with limited abilities. In contrast, in some mammals, excessive apoptosis can hinder regeneration, leading to scar formation instead of functional tissue replacement. Understanding the balance between survival and death of cells is vital. The insights gained from these investigations contribute to a broader understanding of regeneration, with implications for developing innovative therapies aimed at enhancing healing processes in humans. Environmental factors play a crucial role in shaping the regenerative capabilities of various species. Specific habitats exert selective pressures that influence the evolution of regeneration strategies. In aquatic environments, for instance, the presence of predators and the complexity of the ecosystem can dictate the necessity for rapid regrowth of limbs or organs. These adaptations allow organisms to survive and thrive in environments characterized by high predation risk. In terrestrial ecosystems, variations in temperature, moisture, and nutrient availability create distinct challenges that necessitate different regenerative responses. The ability to regenerate may be enhanced in regions where resources are abundant, allowing for quicker recovery from injuries. In contrast, harsher climates may lead to more gradual, energy-efficient regrowth processes. Different species exhibit unique responses to their environments. For example, certain amphibians, which inhabit diverse ecosystems of you, show remarkable limb regeneration abilities that are influenced by their specific surroundings. This variation highlights the intricate relationship between habitat and regeneration. Furthermore, the interplay between biotic and abiotic factors often defines the limits of regenerative potential. The presence of symbiotic organisms, such as bacteria, can also enhance regenerative processes by contributing to the healing environment. Understanding these environmental adaptations provides deeper insight into the evolutionary significance of regeneration across various species. Climate change exerts significant pressure on ecosystems, influencing the delicate balance that supports regeneration. Altered temperatures and extreme weather events hinder the habitats of myriad organisms, affecting their ability to recover from injury or loss. In this shifting landscape, regeneration becomes a critical factor for survival, as species must adapt to rapidly changing conditions. Coral reefs, known for their remarkable regenerative capabilities, exemplify this struggle. Rising ocean temperatures lead to coral bleaching, diminishing their vitality and capacity to regenerate. Disrupted symbiotic relationships with algae impair their ability to recover, resulting in widespread coral loss. This decline ripples through marine ecosystems, threatening species that rely on these structures for shelter and sustenance. Similarly, terrestrial environments witness challenges to regeneration. Forests, home to numerous species capable of regrowth, face increasing frequency of wildfires exacerbated by climate change. This phenomenon alters soil composition and disrupts the regeneration processes, contributing to biodiversity loss. The implications extend beyond specific species. As habitats are transformed, the interconnected web of life becomes increasingly fragile. Species that once thrived may struggle to adapt, leading to cascading effects on food webs and ecological stability. Monitoring regeneration rates and their responses to climate variables becomes essential in understanding the broader impacts on biodiversity. Informing conservation efforts aimed at preserving these vital ecosystems. The evolution of regeneration serves as a remarkable testament to adaptive strategies forged over millions of years. Various species exhibit distinct regenerative abilities, ranging from the modest capabilities of certain amphibians to the extraordinary feats of starfish and axolotls. This diversity in regeneration reflects the intricate interplay between environmental pressures and survival mechanisms essential for thriving in specific habitats. In these regions, the ability to regrow limbs or organs not only enhances individual fitness, but also contributes to population resilience. The genetic foundations underpinning such regenerative capabilities reveal a wealth of cellular pathways and molecular signals. Scientists study these processes to uncover the underlying biological frameworks, providing insights that may one day inform medical advancements in regenerative medicine. Some organisms leverage their regenerative prowess in response to predation or habitat destruction. For example, the regeneration of lost appendages can support prey species in evading predators, while simultaneously allowing for continued participation in ecological interactions. Understanding the evolutionary context of these adaptations enables researchers to predict how species might respond to ongoing environmental changes. Investigating regeneration also sheds light on the limits of these abilities. Not all species can regenerate indefinitely or to the same degree, revealing constraints shaped by evolutionary history. These patterns elucidate the complex relationship between biology and environment, a dynamic shaping the future of life on Earth. Future directions in regeneration research promise to deepen understanding of these biological phenomena. Scientists are exploring the genetic and molecular mechanisms that govern regenerative capabilities by examining species known for their extraordinary healing abilities. Researchers aim to identify specific genes responsible for regeneration. This genetic insight lays the groundwork for potential applications in medicine, particularly in wound healing and tissue engineering. Investigations extend into the biochemical pathways involved in regeneration. It appears that signaling molecules play a crucial role in initiating and sustaining the regeneration process. In this context, the study of these pathways may reveal strategies to enhance regenerative responses in humans, possibly leading to breakthroughs in treating injuries and degenerative diseases. Environmental factors, such as temperature and nutrient availability, significantly influence regenerative processes. Understanding how these variables affect regeneration could guide conservation efforts for vulnerable species. Research in this area highlights the importance of habitat preservation, as the loss of biodiversity may hinder the regenerative potential of ecosystems. Interdisciplinary collaborations are becoming increasingly essential. Combining insights from genetics, ecology, and bioengineering may accelerate advancements in regenerative medicine. The quest to unlock the secrets of regeneration encapsulates a profound intersection of biology and technology, paving the way for innovative approaches to enhance life and health across species. The potential of regeneration extends beyond mere curiosity, reaching into the realm of disease management and treatment methodologies. Research into organisms capable of remarkable regrowth reveals insights that could revolutionize how conditions such as cancer and neurodegenerative disorders are approached. Understanding the cellular processes that enable certain species to regenerate lost limbs or organs may inform therapeutic strategies for humans suffering from similar challenges. This region of study encompasses the mechanisms behind cellular reprogramming and stem cell activation. In species like axolotls, the regeneration process involves a complex interplay of signaling pathways, which, when harnessed, might lead to breakthroughs in regenerating human tissues. The implications stretch further, hinting at the ability to repair damaged organs or restore function after traumatic injuries. Exploration into the genetic frameworks governing these abilities has already begun to yield promising results. Researchers are delving into the roles of specific genes and proteins, which may serve as targets for future treatments. By manipulating these biological mechanisms, it becomes conceivable to enhance the body's innate healing processes, potentially leading to novel interventions for chronic diseases. The relationship between regeneration and health continues to unfold, suggesting a future where the line between healing and regeneration blurs, opening pathways to a more resilient biological existence. Advances in regenerative therapies for age-related conditions are emerging as a promising frontier. Research highlights the capacity of certain cells to not only repair but also rejuvenate tissues that have deteriorated with time. This region of scientific inquiry involves stem cells, which possess remarkable versatility. By harnessing their power, scientists aim to develop treatments that restore function to aging organs and alleviate the symptoms of degenerative diseases. These therapies focus on various age-related conditions such as osteoarthritis, where cartilage degradation leads to chronic pain and reduced mobility. By introducing regenerative cells or growth factors directly into the affected joints, it is possible to stimulate the body's natural repair mechanisms. Results from clinical trials indicate significant improvements in both pain management and mobility in patients following such treatments. The exploration of gene therapy is also underway, offering insights into correcting cellular dysfunctions that contribute to aging. By targeting specific genes involved in cellular regeneration, it may be possible to reverse aspects of the aging process itself. These interventions hold the potential to transform not only the treatment of age-related illnesses, but also to enhance overall vitality. Future research will continue to refine these techniques, paving the way for therapies that could redefine aging. The implications of these advancements extend beyond individual health, influencing societal perceptions of aging and longevity. Cultural narratives surrounding regeneration often intertwine myth and reality, shaping collective understanding of life cycles. In many traditions, the ability to regenerate is attributed to deities or extraordinary beings, reflecting deep-seated desires for immortality and renewal. These stories serve as metaphors for resilience, illustrating the human aspiration to overcome mortality. Ancient texts frequently feature creatures capable of remarkable recovery, such as the phoenix, which rises anew from its ashes, symbolizing rebirth. This imagery resonates across civilizations, emphasizing a universal fascination with the concept of restoration. Legends often depict healing powers bestowed upon certain animals, suggesting a connection between nature's wonders and human longing for invulnerability. Scientific inquiry into regeneration offers a contrasting perspective, grounded in biological mechanisms rather than mythological interpretations. Research reveals that organisms like salamanders and planarians possess intricate cellular processes, enabling tissue regeneration. These findings challenge long-held beliefs about the immutability of aging and injury, prompting a re-evaluation of natural limits. In society, contemporary society, narratives surrounding regeneration are evolving. The intersection of science and technology introduces potential for therapeutic applications, raising ethical questions and societal implications. Discussions about regenerative medicine often evoke reflections on identity, mortality, and what it means to be human. These dialogues reflect an ongoing quest to understand the boundaries of life, informed by both ancient law and modern science. The integration of citizen science into regenerative research has emerged as a pivotal development in the study of organisms capable of remarkable regeneration. Community involvement offers an expansive array of data, often surpassing what traditional methods can achieve. Local enthusiasts, equipped with curiosity and basic tools, contribute invaluable observations and insights, enhancing understanding of regenerative processes across diverse species. In these collaborative efforts, volunteers document their findings, which can range from simple anecdotal records to detailed phenotypic measurements. This grassroots approach democratizes scientific inquiry, allowing individuals from various backgrounds to engage meaningfully with complex biological phenomena. The collective knowledge generated fosters a deeper appreciation for the nuances of regeneration, broadening perspectives beyond academic circles. Field studies benefit significantly from this participation, as citizen scientists monitor habitats and track the health of populations over time. Such observations can elucidate how environmental factors influence regenerative capabilities, revealing patterns that may go unnoticed in isolated laboratory settings. Furthermore, these contributions often lead to the discovery of new regenerative species, enhancing biodiversity records and potentially guiding future research avenues. In essence, the involvement of the public in scientific endeavors not only accelerates research, but also cultivates a sense of stewardship. This connection between people and nature encourages the preservation of ecosystems where regenerative talents thrive, securing a future for these remarkable organisms. Future technologies in regenerative medicine promise to harness the inherent capabilities of organisms known for their regenerative prowess. Advances in biotechnology are paving the way for innovative treatments that could mimic these natural processes in humans. Researchers are exploring gene editing techniques, particularly CRISPR, to modify human cells and enhance regenerative abilities. These developments may allow for the reconstruction of damaged tissues and organs, offering solutions for ailments that currently lack effective treatments. Stem cell research is at the forefront, with potential applications ranging from wound healing to the regeneration of complex structures like hearts and limbs. The manipulation of cellular pathways is revealing how certain organisms can regenerate, guiding scientists in replicating these processes artificially. Biomaterials are also gaining attention, with scientists developing scaffolds that support tissue growth. These materials could be combined with cells to facilitate healing and regeneration in a controlled manner. The integration of 3D printing technology further enhances this field, allowing for the creation of custom prosthetics and tissue mimetics tailored to individual patients. Ethical considerations accompany these advancements, necessitating thoughtful dialogue as the capabilities of regenerative medicine expand. The intersection of nature and human ingenuity holds the potential to redefine medicine, fostering a future where lost tissues and organs can be restored and functionally integrated into the human body. Building such a regenerative society requires a comprehensive understanding of biological processes and their applications in community health. This concept extends beyond individual restoration, encompassing the collective well-being of populations. Local ecosystems play a pivotal role, as they provide resources and environments that foster healing. Communities that prioritize regenerative practices create spaces for collaboration, sharing knowledge about sustainable living and health. In these environments, the exchange of ideas enhances both scientific research and community resilience. Education becomes a cornerstone, empowering individuals to comprehend the mechanisms behind regeneration. Workshops and outreach programs cultivate awareness about the remarkable abilities of certain species, such as starfish and axolotls, serving as biological blueprints for innovation. These initiatives promote not only physical healing but also social connection. The act of gathering to explore these principles strengthens bonds among community members, fostering a shared purpose. Furthermore, the integration of regenerative medicine into healthcare systems encourages holistic approaches. It emphasizes the importance of nurturing both body and community, recognizing that healing is a multifaceted process. Regeneration plays a critical role in maintaining the balance of ecosystems. This process allows various species to recover from disturbances, whether they are natural, such as wildfires, or human-induced, like pollution. Through regeneration, flora and fauna can restore their populations and adapt to changing environments, promoting resilience within their habitats. These ecosystems depend on a delicate interplay between organisms, where each species contributes to the overall health of the community. The regeneration of certain plants can provide food and shelter for various animal species, while the return of these animals helps in seed dispersal and nutrient cycling. This reciprocal relationship fosters biodiversity, which is essential for the adaptive capacity of ecosystems. The ability of certain species to regenerate not only enhances their survival, but also serves as a buffer against ecological collapse. In areas where regeneration is stunted, the consequences can be dire, leading to diminished resources and increased vulnerability to invasive species. The decline of even a single species can ripple through the entire ecosystem, highlighting the interconnectedness of life. Understanding the mechanics and significance of regeneration invites a deeper appreciation for preserving natural habitats. Conservation efforts aimed at protecting regenerative processes ensure that ecosystems retain their integrity and continue to thrive, fostering a vibrant web of life. The potential for regeneration extends beyond the natural world, inviting exploration within the realms of science and medicine. Groundbreaking research reveals how understanding these processes may lead to revolutionary advancements in tissue engineering and regenerative therapies. Insights drawn from species capable of remarkable regrowth open avenues for innovative treatments in humans, particularly in the repair of injuries and degenerative diseases. This pursuit has led to the development of biomimetic materials and techniques that seek to replicate the intricacies of natural regeneration. Scientists harness the mechanisms found in organisms like axolotls and planarians, studying cellular reprogramming and the role of specific genes in regeneration. These discoveries may one day provide solutions for conditions currently deemed irreversible. Laboratories worldwide are investigating the implications of stem cell technology, exploring the way these versatile cells can be guided to develop into various tissues. This research holds promise for restoring function in damaged organs, presenting a future where ailments such as heart disease or spinal cord injuries may be treated with ease. As the quest for regenerative solutions continues, the boundaries between biology and medicine become increasingly blurred. With each breakthrough, the vision of a world where healing is not only possible but also extends life itself becomes more tangible, illuminating a pathway to endless possibilities in the realm of health and healing.